guys, so I'm going to try something a little bit different. I've been working with a few of my IT people and trying to figure out what's going to be the best experience for you guys. And I know that you guys have my PowerPoints, and I know that for the first couple of lectures you were kind of seeing my face right there in your screen. And then I came across a couple of things where maybe I can do it both. So I'm going to try and switch it up a little bit so you guys don't get bored. That's one thing I definitely don't want you guys to be. I want you to get the most out of this class. I told you guys the other day, this is a fantastic class, and I genuinely do mean that. So for a little bit, we're going to just try to do this where you can see my PowerPoint, and you can see me over there in the corner. How are you? And we're going to move right along into talking about some of the differences between prokaryotic species and eukaryotic species. They're easy really to compare when you take a look at them kind of side by side. I realize you can't see the entire slide, and I'm sorry, this is kind of the biggest that my screen will get, but you do have the PowerPoints there with you, so you'd be able to fill in that top portion that you can't see. But with prokaryotes and eukaryotes, you guys learned eukaryotic species when you took anatomy and physiology. You talked about how they divide through mitosis. You talked about the fact that we as humans don't have a cell wall. We have a plasma membrane, but we don't have a cell wall. Plants, fungi, they have a cell wall. Eukaryotes have organelles, and each of those organelles have a different job. The ribosome, the mitochondria, the endoplasmic reticulum, the Golgi apparatus, things like that. We talked about chromosomes and the fact that they have a paired amount of chromosomes that are found within a nucleus. When you look over at the prokaryotic side, though, prokaryotics are very different. Their species divide much differently. We talked about that binary fission where they copy their DNA and then they split into two identical types of species, two identical cells. We talked about the fact that they do have a cell wall, and it's an extremely complex cell wall that does have peptidoglycan in it. They don't have any type of organelles. Some of the questions that I sometimes ask people will say, one of the answers will say, you know, on the back or within the bacterial mitochondria, and I have people check that. And I'm like, why? Why are you checking that? You know that they don't have a mitochondria. So don't get tripped up like that. They don't have any type of mitochondria, no Golgi apparatus. They don't have any histones. They do have one single circular piece of DNA that is not enclosed in any type of nucleus. There's no membrane that is around it. When you look at prokaryotic cells, and we're going to focus for the most part on prokaryotic cells because that is going to be the that's going to be the main focus of the class in this chapter. So when you look at prokaryotic cells, you do see some of those basic shapes that I was talking about back in chapter 3. Take a look up there. That one says diplococcus because the word diplo means two, and those are two circles side by side, which would be by definition a diplococcus. You then see some of those bacillus. There's one of those spirochetes down here in the corner. Can I point to it? Oh, right there, down there in the corner. It's that neon green spirochete. So time out. That is a fluorescent microscopy. That is a neon green. That is a spirochete. You tell me, what is that species? That is syphilis. That's going to be that species, Treponema pallidum, that we were talking about back in chapter 3. Most of the bacteria that we are going to study don't change their shape. They are single shape. Some of these more unusual type of shapes, those are going to be saved for our archaea. When we look at microbes, they are arranged differently. I do want you to know the arrangements because when you get here in lab, it's going to be important that you be able to identify what these microbes are looking like. So by definition, the word diplo means two. So if I have diplococcus, that's going to be two circles side by side. If I have diplobacillus, what do you think that's going to look like? That's going to be two rod-shaped side by side. The word staphylo means cluster, clumped together. You remember when you were a little kid, you drew grapes? Well, I still draw them like this, but you drew like circles all clustered together for a bunch of grapes. That's exactly what Staphylococcus looks like. They're a bunch of clumped together circles. And then we have Streptococcus and Streptobacillus. The word strepto actually means chain. So if you have a chain of rods or if you have a chain of circles, that's going to be Streptococcus and Streptobacillus. Some of the things that we find in prokaryotic species that are not necessarily unique to them, because there are glycocalyx that are going to be talked about a little bit in eukaryotes as well. But some of the things that prokaryotes have, they make this inside of the cell, and then they secrete that to the outside of the surface, and usually it's a sticky type of substance. This is the glycocalyx, and the glycocalyx sometimes comes very, very organized. If that is the case, that is a capsule. You guys heard about a capsule just in the last lecture. I talked about how we stain for capsules. I talked about the fact that they're really, really difficult for our immune system to find because they're protecting. It's like a bubble that protects those microbes. Some of the species that have these capsules, Bacillus anthracis, Streptococcus pneumoniae, Klebsiella. Funny story about Klebsiella. I will never, ever live down the first time I ever saw Klebsiella. I was a second-year medical student, and I was working in a clinic that was the downtown clinic. It's a free clinic in Louisville. And a gentleman came in who was often there. He was homeless. He was a known alcoholic, known drug abuser, who came in and he had this kind of rattly sounding cough. It wasn't any surprise that he was there. He was always there. But I went in and was talking to him, asking him how he was feeling, and took his temperature, and he did have a fever. Well, all of a sudden, he coughs, and up comes this hunk of, oh, it was blood, it looked like grape jelly. Like if you took a big old spoonful of grape jelly and globbed it in the middle of your hand, that's what it looked like. And in my head, all I could think about was, he's dying. He is hemorrhaging. He has hacked up half of his lungs somehow. And so I did what, any good medical student would do, and I ran. Number one, friends, don't run from a patient. You should never run. But I ran. I took off and I ran, and I found an attending, and we go running like an episode of of ER Grey's Anatomy. We're running, and we like push through the double doors, and we enter into the room, and he walks in, and he took, he looked at the patient, he looked at me, he looked at the patient, and he looked at me like I was the dumbest person in the world, and he said, "Did you even bother to read your textbook?" And I was thinking, no, I didn't. So then he started going over some things with me. Known alcoholic, recurrent types of infections, current jelly type sputum, and all of a sudden it clicked with me that that is textbook Klebsiella. So Klebsiella pneumoniae is a pneumonia that not just alcoholics get, but they are more likely to get Klebsiella. We do see them being more kind of subject to that. Another type of glycocalyx that we find sometimes is made of sugars, and actually bacteria will actually draw glucose and sugars from their environment, and that's what Streptococcus mutans do, and they live in your mouth. Your mouth is disgusting. There are more microbes in your mouth than you could ever possibly imagine. But inside of your mouth, these Streptococcus mutans will live amongst your gum line, down in between your teeth, and they're collecting sugar anywhere where they can get it. And they build up this glycocalyx around them that helps to protect them. And then what happens is as they're being protected by this glycocalyx, the microbe starts to kind of break through and break down the enamel in through the dentin and on into the pulp, and that's what a dental caries is, which you guys probably call a cavity. So that's why when your mom used to say, you know, candy causes cavities, well, not technically, candy doesn't cause cavities. What causes cavity are bacteria. And when that bacteria has all that candy, what's present? All that sugar. And so what we find is that those microbes are going to take that sugar and they're going to wrap that in their glycocalyx and really use that against you. So that's why it's really important to make sure you brush your teeth, make sure you floss really well to get all that out of there so they can't have it. We talked about the fact that some microbes can move, and when they move, they're able to use flagella, and we talked about the stain that we have to do that. The flagella, the way that they work, they actually have this anchor, if you will, that is inside of the basal membrane, and they have a protein hook that hooks into, and then that cranks and turns and therefore whips the flagella along so they're able to move. Not all microbes have flagella, and not every single microbe with a flagella looks the same. So what we find is that flagellar species come in different arrangements. If you only have one single flagella that kind of sticks out of the end, that's called monotrichous. If you have one single flagella, but it's at one end and one at the other end, that's called amphitrichous. Some of them still at the polar ends, but they have multiple ones that come out. That's called lophotrichous. And then some microbes have what are called peritrichous, and they have these flowing flagella kind of all over their bodies. The way that they do work, though, as I said, they have these pairs of rings. Gram-negative have two pairs of rings. Gram-positive only have one set of rings. They only have the inner set. And what they do is they crank and they turn and they rotate that flagella for movement. When bacteria move, they move in a special way called run and tumble. So they essentially run in one direction, and then they do a flip, which is called the tumble, and then they run in a different direction, they tumble, and they run, and they tumble, and they run, and they tumble, and they run. And we used to think that this was completely random. We now know that that's not actually true. It's not necessarily something that is random, and what we have found is that when we have this this movement, sometimes it's actually on purpose. We can manipulate microbes to move in a process called taxis. Let's say, for example, that over here, make sure my fist is where you can see it. Okay, so over here we have something that would be of benefit to the microbe. Microbes love iron. They scavenge iron. So let's say this is iron, and over here is a microbe, and it can actually sense with receptors on that cell wall that this iron up here is actually going to be good for it. And so what happens is that microbe will tumble and make itself run in a pathway that is going to actually come into contact with that iron, so it can then uptake it and utilize that. So we used to think it was completely random, and now we know that it's not always a random process. There is an exception to the rule. I feel like there's always kind of an exception to the rule, but the exception to the rule is the spirochete. Spirochetes have a unique way of moving because, number one, of their shape. They have that kind of, wavy-like of shape. And they have what's called an endoflagellum. So that means that anchored at one end of their body, it runs internally and it turns. So since they're already shaped in that spirally type of movement, when they rotate, they have that sort of corkscrew movement as opposed to the run and tumble. We have found several different interesting things about how microbes are able to latch on and colonize our body. When microbes get into your body, if they're going to cause any type of damage, they have to find some way to attach. And that attachment is going to be necessary for their survival. Neisseria gonorrhea. If you have, oh, you got the gonorrhea. Gonorrhea. Becoming, you know, when I first started with, I told you guys, or I may not have told you guys, I have a huge obsession with STDs and parasites. But when I started an OB rotation, I was a little bit taken aback by just how many people are positive for STDs, and gonorrhea and chlamydia are everywhere. Everywhere. So Neisseria gonorrhea, the vaginal cavity is actually very difficult for microbes to really colonize. Number one, it's warm, and microbes don't really like that warm of temperature. Number two, it's acidic. Microbes, for the most part, don't really like acid. And number three, there's constant mucus and secretions that are being made, which makes it difficult to colonize. Neisseria gonorrhea, that does cause gonorrhea, has found an excellent way because they have what are called fimbriae. Fimbriae are these hair-like attachments that stick off of the microbe. They're not flagella, they're not for movement, they're for attachment, and they're very sticky, and they allow those species to colonize places that might be a little bit difficult when it comes to colonization. Another interesting thing that microbes have that we're going to be covering really in detail in Chapter 8 are called pili. Sometimes I like to think about the whole concept of, it's kind of like that sci-fi concept, what if we could do what microbes could do? Pili would be one of those that are really, really fascinating, simply because with pili, what happens with that is microbes are able to isolate DNA, first of all, copy that DNA, second of all, and pass that DNA, transferring it from one microbe to the other. Now, I'm not talking about from offspring. I'm not talking about from mother to offspring. That's not what I'm talking about. I'm not talking about mother to daughter cell. I'm talking about the same generation cells. For example, my husband's name is David. He's very sweet. I showed you a picture of my kids. Oh, I don't have a picture of David. Whoops. Okay, don't anyone tell him that. I don't have a picture of him in here, but I showed you a picture of my kids, and so I'm going to tell you a little bit about David. He's been my best friend since I was about 7 years old. So we've known each other for a very, very long time, and I have always loved his eyes. He has these green, piercing eyes that are just fantastic. And let's say that we were microbes and we could do what microbes do. So I go up to David and I say, hey, honey, because if I want something, yeah, I call him honey, sweetness, sugar lips. I was just wondering if I could have your eye color. And he would say to me, absolutely. And he could internally isolate the gene for eye color, make a copy of that, and then we would form this pilus between us. It's a direct connection between us, and that copy of his eye color would travel through the pilus and into me. I would be able to uptake that genetic information into my own genes, close my eyes and open them, and I would have his eye color. That's mind-blowing. Can you imagine? Now, of course, we can't do that. We can't transfer DNA between same generation cells, but microbes can. Bacteria can. Bacteria don't have beautiful green eyes, though, but they do have things like antibiotic resistance, and they do have things like toxin production. So think about this. Microbes are not selfish. They are not stingy. If I'm a microbe and you're a microbe, and I see that I am penicillin resistant, which means that penicillin doesn't do anything to me. But you are penicillin susceptible, which means that it will kill you. I can isolate that gene for penicillin resistance, and I can copy that, and I can form that pilus and transfer that right into you. So now you and me are both antibiotic resistant. Microbes are really, really fascinating. I'm going to pause right here for a second. We're going to pick up with the cell wall. And I told you guys that the cell wall is going to be where we figure out and find out just how different gram-positives and gram-negatives are. So I'm going to upload this for you guys to have, and we'll be back in a bit.